Hello all, today's topic is DNA replication. DNA is deoxyribonucleic acid. DNA replication, as a name suggests, replication means making its own copies. So DNA is self-replicating structure, and this DNA replicate, it means it makes multiple copies of itself. And DNA replication is a semi-conservative type of replication. So, in DNA replication, there are three important processes that involve. The first one is initiation, then elongation, and in the end termination. So, in DNA replication, the first step is initiation. Initiation. So, DNA replication demands high amount or high degree of accuracy. So, uh, any high degree of accuracy, even minute changes that can occur in DNA replication can cause a mutation. That mutation can be life threatening or can be severe as well. So it is really important to initiate the DNA replication properly. So DNA replication cannot be initiated randomly at any point. For replication to begin, there is a particular point for DNA replication, and that point is called as origin of replication. So here it is. This is a region where origin of replication started. So here is replication originate. So replication begin by spotting the origin, and then it unwind the two strands of DNA. And unzipping of DNA structure in the entire length is not feasible because it needs high energy. So because of the high energy input, it is not possible. It is not feasible. Hence, the first replication fork is formed, and this is created by helicase enzyme. So helicase enzyme play very important role in unwinding of the DNA. After unwinding, there comes an elongation. In elongation, as a strand separate, the next enzyme that come in action is polymerase enzyme, DNA polymerase 3. It starts synthesizing complementary strands in each of the strand. So parent strand act as a template, and it make a daughter strand. So elongation is unidirectional, that is from 5' to 3' direction, and while on other end it is discontinuous because it start with 5' to 3', but elongation happens in 5' to 3' direction only. So the one that is 3' to 5' direction, the elongation is continuous, while the another is discontinuous. So this forms a fragment, and that is called as Okazaki fragment. And these Okazaki fragment are stick together by the enzyme called as DNA ligase. So DNA ligase helps in sticking of this Okazaki fragment and make a continuous form of DNA. After making this Okazaki fragment are stick, then comes an end of this replication. That replication is called as termination. So in termination, the termination occurs in different way for different organism. So suppose in E. coli, chromosome are circular. So thus it is happen when two forks meet each other, and then DNA termination means DNA replication termination happen. So there is a TUS protein. TUS protein is terminus utilization substance, and they binds to the terminating sequences and halts the DNA polymerase movement. DNA polymerase movement is halted by this terminal, and this way DNA replication has stopped. So DNA replication is occurred in nucleus in eukaryotic cell and in nuclear region for prokaryotic cells. And DNA replication is important because cell must replicate their DNA before they divide. So this ensured that each daughter cell get a copy of the genome, and therefore successful inheritance of genetic triad. So this will help to maintain the genetics of the parent cell as well. So DNA replication is essential process, and the basic mechanism uh, is conserved in all organisms. So therefore DNA replication is really important. DNA replication is are of two types. First is conservative DNA replication. In conservative DNA replication, it produces two DNA helices, out of which one contain entirely old DNA and one contain entirely new DNA. And second one is conservative type of replication. So it produces DNA helix in which each helix contain new strand and old strand, and therefore DNA replication in humans is semi conservative. This is all about DNA replication. Thank you.